imagine, just for a second, a medical future with, like, absolutely zero organ wait lists.
Right, yeah. That's the dream.
A future where you're lying in an ICU, you know, your liver and kidneys are shutting down, multi-organ failure is setting in, but you don't have to wait for a human match to be found in time.
You just get what you need.
Exactly. The organs are just available, ready to go, engineered specifically for human biology.
I mean, it is the ultimate holy grail of transplant medicine. For decades, researchers have been chasing this exact concept and mostly just running into, well, insurmountable biological walls.
Because our immune systems are a little too good at their jobs.
Yeah, exactly. The human immune system is just incredibly efficient at recognizing and destroying anything that, isn't supposed to be there.
But a groundbreaking 2026 paper published in the journal Med suggests we might have finally breached that wall.
It's a huge step.
It really is. So today's deep dive is into the staggering medical milestone. We're looking at the first ever orthotopic multi-organ xenotransplantation. We're talking about a whole liver and bilateral kidney, so both kidneys, taken from a highly genetically modified pig and transplanted into a human decedent.
And, the sheer scale of the achievement here cannot be overstated. We aren't looking at, like, a single organ temporarily hooked up to a patient's leg to filter blood for a few hours.
Which we've seen before.
Right, we've seen it. But this is massive, system-wide integration inside the human abdominal cavity.
And our mission today is to go cell by cell, metabolite by metabolite, into the 106 hours following that surgery. We really want to understand exactly how this works.
Yeah, we're not skipping any details today.
No, we're getting into all of it.
Yeah.
So to set the stage, let's look at the clinical reality the team actually faced. The recipient was a 53-year-old male patient.
Tragically declared brain dead.
Yeah, brain dead after a severe brain hemorrhage. But complicating matters, he also had pre-existing stage 4 chronic kidney disease. So his blood work was, it was dire.
Yeah, very bad.
His creatinine levels were sitting at 513 micromoles per liter with highly elevated blood urea nitrogen.
Which, just for context, normal creatinine is usually under 110.
Right.
So level of 513 indicates that his kidneys had essentially just stopped filtering waste. In a living patient, that degree of toxicity means, an immediate, non-negotiable need for dialysis.
Wow. And his family, in just an incredible act of generosity, agreed to participate in this study to advance the science of transplantation.
It's truly amazing that they did that.
It really is. So to solve the failing organ issue, the medical team turned to a very specific donor, a 427-day-old Bama miniature pig.
But this brings up the core mechanical problem, right?
Yeah. If you put 3 massive organs from another species into a human body, the human immune system is going to recognize that foreign tissue instantly.
Instantly. Which is why the procedure, formerly called an orthotopic xeno-CLKT, relies entirely on the genetic engineering of that donor pig.
Okay, let's break that term down for the listener. Xeno means cross-species, obviously.
Right. And the orthotopic part is crucial. In previous experiments, we've seen heterotopic or auxiliary transplants where a pig organ is essentially just tucked alongside the patient's own failing organs.
Like a backup generator.
Exactly. But here, they completely remove the failing organs and place the pig liver and kidneys in the exact normal anatomical position.
And the CLKT part.
That's combined liver-kidney transplantation. They went for a total replacement to achieve integrated systemic homeostasis.
Because, the liver and kidneys aren't just isolated filters, right? They rely on each other.
Oh, heavily. The liver manages metabolic and synthetic functions, and the kidneys handle the excretory balance. If they aren't hemodynamically positioned correctly, the whole system just crashes.
Okay, so let's dig into the genetics of this Bama miniature pig. Because the donor was engineered with 6 very specific gene edits. 3 knockouts and 3 insertions.
I like to use a nightclub security analogy for this.
Oh, I love that. Let's hear it.
Okay, so let's start with the knockouts. Yeah. GGTA1, CMAH, and B4GOLNT2. These 3 genes produce pig-specific carbohydrate antigens.
Basically sugars.
Right, sugars. Think of these sugars like giant glowing neon signs on the surface of every single pig cell, basically screaming, "I am a pig."
Okay, neon signs. Got it.
Human antibodies are like the nightclub bouncers constantly patrolling the bloodstream. If we hadn't knocked those genes out, the bouncers would see those neon alpha-gal sugars, recognize them as foreign, and, initiate an immediate catastrophic attack.
Right, so they essentially removed the neon signs so the human antibodies just glide right past the cells without sounding the alarm.
Exactly. But taking down the signs isn't enough because the human complement system is still a major threat, which brings us to the 3 insertions.
Okay, so HCD46, HCD55, and HTBM.
Right. So the complement system is this cascade of proteins in the blood that, when activated, literally forms a structure called the membrane attack complex, or MA.
And MA punches physical holes in the walls of foreign cells, right,
Yeah.
causing them to burst.
Yeah, it's brutal. So by inserting HCD46 and HCD55 into the pig's DNA, the organs express human proteins that act as a VIP pass.
A molecular handshake.
Exactly. When the human complement proteins dock with the pig cell, those inserted human genes signal back, hey, I belong here, stand down, which disarms that attack cascade.
And that third insertion, HTBM, is human thrombomodulin. I imagine that's there to stop the human blood from instantly clotting the moment it touches the unfamiliar inner lining of the pig's blood vessels.
You nailed it.
Yeah.
Endothelial incompatibility often leads to massive coagulation. So the HTBM insertion smooths that interaction, keeping the blood flowing freely through the new vascular networks.
It's just biological hacking at an astonishing level. And to actually execute this, the surgeons used what's called the sun technique.
Which is fascinating surgically.
Yeah, they made a single upper abdominal inverted T incision. This allowed them to simultaneously perform a nephrectomy, so removing the patient's failing kidneys, while implanting the pig liver and both pig kidneys all at once.
Yeah, this elegant surgical workflow minimized the ischemia time, meaning the organs were without blood flow for the shortest possible window.
Okay, so let's just quickly recap that for you, the listener, before we move on. Bullet 1. We have a brain-dead human recipient with failing kidneys.
Bullet 2. A donor pig, genetically masked with 6 specific edits to evade human immunity.
And bullet 3. A single incision surgery replacing 3 vital organs all at once.
Right.
So here's a question for you to think about. If you woke up with 3 organs from another species, what is the absolute first thing your immune system would try to do?
Well, it brings us to the moment the clamps are removed. Human blood rushes into these newly installed pig organs. The absolute first hurdle your body has to clear isn't long-term survival.
Right. It's like the next 5 minutes.
Exactly. Your immune system is going to try to trigger hyperacute rejection.
HAR. This is literally the nightmare scenario for transplant surgeons.
It really is. Even with the genetic barcode scrubbed and the VIP passes in place, HAR can still occur if pre-existing antibodies find a target.
And that leads to rapid microvascular thrombosis, right? Like millions of tiny blood clots that just suffocate the organ.
Yeah, you do all this work and the tissue just turns black and dies right there on the operating table within minutes.
Terrifying. So to prevent that cascade, the medical team deployed a very specific immunosuppression algorithm. And what's wild is that they largely followed standard allogeneic principles.
Meaning the same protocols used for normal human-to-human transplants.
Right. They administered rituximab 3 days prior to surgery and again on the day of the transplant.
Which completely wipes out the B cells.
Mm-hmm.
And if the B cells are gone, the body can't produce a new wave of antibodies specifically tailored to attack the pig tissue.
Okay, makes sense. And then they also used tacrolimus to suppress the T cells, but they kept the dose surprisingly low.
Very low. The target trough was just 5 to 8 nanograms per milliliter, which is about 1 milligram per liter in practice.
That feels incredibly low. I mean, previous studies that just hooked up a piece of a pig liver to a human required massive doses of tacrolimus, sometimes around like 5 milligrams per liter.
Yeah, much higher.
So why did they get away with so little immunosuppression here? Is it because they transplanted 3 massive organs at once? Like, does a whole liver and 2 kidneys act like a giant immunological sponge?
That is actually the leading hypothesis right now.
Yes.
The sheer volume of the transplanted tissue might induce a form of early immunological tolerance.
Oh, wow. So it just absorbs whatever circulating antibodies are left and spares the system from a concentrated attack.
Exactly. It dilutes the autoimmune response across a much larger vascular surface area. And they paired this with mycophenolate motile and corticosteroids to keep the baseline systemic inflammation in check.
And the clinical markers of success were immediate. Let's look at the liver function first. At 19 hours post-transplant, the pig liver secreted 1.5 milliliters of bile.
And by 48 hours, that volume rose to 20 milliliters.
Which is huge, because producing bile is like the ultimate green light for a transplant surgeon, isn't it?
Oh, absolutely. It's not just about fluid moving through a tube. Bile production definitively proves 3 physiological facts.
Okay, what are they?
First, hepatocellular synthetic activity. The liver cells are actually alive and synthesizing complex molecules. Second, biliary patency. The structural plumbing of the organ is intact.
Right.
And third, microcirculatory perfusion, meaning blood is successfully reaching the most microscopic vessels of the organ to fuel that cellular activity.
And the imaging backed this all up. Ultrasound confirmed the portal vein flow was a very robust 15 to 20 centimeters per second.
Which is great.
Yeah, and the blood tests showed exactly what you'd expect. AST liver enzymes spiked initially from the physical trauma of the surgery, but then they fell naturally.
While ALT, which is a highly specific marker of acute liver injury, stayed perfectly within the normal range.
But the real revelation came from the kidneys. Remember the patient's baseline creatinine, a highly toxic 513.
Right. His native kidneys were just completely failing.
But after the pig kidneys were perfused with human blood, that creatinine level plummeted all the way down into a normal human range. The urine output stabilized.
The pig kidneys were flawlessly filtering human blood and pulling the toxic waste out of the system.
It's amazing. And as they were monitoring the blood chemistry, they uncovered this brilliant unintended bonus mechanism. The patient's uric acid levels dropped significantly as well.
Yeah, this part is fascinating.
Wait, human livers don't produce the enzyme uricase? Right. Which is why we struggle to break down uric acid and why people get conditions like gout.
Exactly. But pigs naturally produce uricase.
So does this mean the pig liver was actively metabolizing human waste that our own native livers can't even process?
That is exactly what was happening. The pig liver was pumping out porcine uricase, aggressively clearing the human bloodstream of uric acid. It was functioning beyond standard human capacity.
But okay, that raises a major red flag for me. If this pig liver is pumping out pig proteins and pig enzymes into the human bloodstream, wouldn't the human immune system eventually detect all that circulating foreign material and mount like a delayed attack against the organ?
Yeah, it's the logical assumption. So to monitor this, the researchers utilized detailed proteomics. They studied the exact proteins circulating in the plasma over those 106 hours.
And what did they find?
They found something extraordinary. In the initial hours, there was a high concentration of porcine albumin, pig proteins, in the blood. But over time, that porcine albumin progressively decreased.
Okay.
And it was replaced by the human isoform.
The human isoform, wait, meaning the organ was assimilating?
Essentially, yes. A form of functional chimerism was taking place. The pig liver was utilizing human substrates from the blood to synthesize proteins, gradually shifting its output to match the human biological signature.
That is mind-blowing.
Right, it was biochemically adapting to its new host, reducing the amount of pig-specific immunogenic material it was putting into the bloodstream.
Okay, let's do another quick recap for you guys. Bullet 1, HIR, hyperacute rejection, was successfully bypassed, and no microthrombi appeared until 36 hours.
Bullet 2.
Okay.
The liver produced bile and pig-specific enzymes, like uricase, that actively help the human body.
And bullet 3. The kidneys successfully filtered the blood, restoring normal creatinine levels.
So here's another question for you, the listener. We know the gross anatomy is working perfectly, but what is happening at the microscopic single-cell level?
Right, because to map that microscopic battlefield, the researchers deployed single-cell RNA sequencing, or ccRNA-seq.
Which is an incredible technology.
Yeah, instead of just looking at a chunk of tissue and guessing what's happening, cRNA-seq isolates individual cells and reads their specific RNA transcripts.
It tells you exactly which genes that specific cell is turning on or off at that exact moment in time.
It's like reading the private diaries of millions of individual cells, taking a cellular roll call.
That's a great way to put it. And when they read the diaries of the cells in the blood, liver, and kidneys at precise intervals, 0 hours, 2 hours, 6 hours, they saw the human innate immune system mobilizing.
Specifically the neutrophils, the shock troops.
Exactly. Within hours, human immune cells were swarming the pig organs, and the data showed a massive, highly specific expansion of one subtype, S100A12 positive neutrophils.
Let's break down what S100A12 actually is, because these cells were clearly running the show.
So S100A12 is a calcium-binding protein that acts as a potent driver of pro-inflammatory signaling. When neutrophils express this protein, they become central communication hubs.
They coordinate the whole innate immune response.
Right, dictating how aggressively the body reacts to the foreign tissue.
And they were coordinating with other specialized cells, right? The sequencing picked up F13A1 positive neutrophils, which handle the complex crosstalk between the immune system and the blood coagulation pathways.
Essentially deciding whether to initiate microscopic blood clots around the foreign tissue.
Right. And there were also PPBP-positive neutrophils, which heavily influence platelet interactions.
And all of these cells need a central command signal. The RNA transcripts reveal the master switch, the ADGRE signaling pathway.
We'll say ADGRE.
Yeah, when the human body detected the massive biological structure of the pig organs, it activated the ADGRE pathway in these neutrophils. This pathway drives two major downstream molecular effects. First, it triggers PI3K-AKT,
Which is a signaling cascade that forces the cells to rapidly multiply and proliferate, right?
Correct. And second, it triggers NFKB, a protein complex that controls the transcription of DNA, essentially forcing the cells to release a flood of cytokines.
The chemical alarm bells of systemic inflammation.
Exactly.
Okay, but let's look at the reality of those mechanisms. If you have all these human neutrophils flooding into the pig liver and kidneys, aggressively multiplying via PI3K-AKT, and screaming for inflammatory backup via NFKB, shouldn't the cellular tissue of those organs be turning into necrotic soup?
You'd think so.
It just sounds like an overwhelming immune invasion.
And if you only looked at the RNA data, you would assume the organs were being destroyed cell by cell. But the physical histology told a completely different story.
The microscopic examination of the actual tissue.
Right? Pathologists took biopsies of the organs and used standard H&E staining, hematoxylin and eosin, to visually assess the cellular architecture.
And H&E staining would clearly highlight massive necrosis or structural collapse if the immune cells were actually shredding the tissue.
Exactly. But they didn't find that. The basic architecture of the liver and kidneys remained remarkably intact. There was no widespread tissue death.
Wow.
And to understand why, they used another marker called Ki-67.
Oh, Ki-67. That's a protein that is strictly associated with cellular proliferation. It only lights up under a microscope when a cell is actively dividing and multiplying.
Exactly. The Ki-67 stains revealed that the cells inside the pig liver and kidneys were highly active, but they were proliferating, they were regenerating.
They're fighting back.
Yes. The organs were taking the inflammatory hit from the human neutrophils and actively repairing themselves in real time, holding their ground against the influx.
We even saw this at the functional genetic level, when they checked the specific genes responsible for the organ's primary jobs, like AQP1 and AVPR2 in the kidneys.
Which code for the crucial water channels that allow the kidney to concentrate urine.
Right, those genes remained fully active and stable. The organs were under siege, but their core functional machinery never went offline.
And this brings us to what might be the most profound discovery of the entire study. If these organs are surviving the immune siege, repairing themselves, and processing human blood over several days, what does that do to the overall metabolism of the human body?
Okay, let's do recap number 3 before we answer that. Bullet 1. Single-cell sequencing showed human immune cells infiltrating the pig organs immediately.
Bullet 2. S100A12 positive neutrophils were the ringleaders, controlled by the ADGRE pathway.
And bullet 3. Despite the immune invasion, the organs did not suffer massive necrosis. They actively repaired themselves.
So the question for you to ponder now is, with a pig liver processing human blood, what happens to the overall metabolism of the body? Does the body become more pig, or does the organ become more human?
To answer that, the team turned to untargeted metabolomics. They took samples of the bile, plasma, urine, and tissue and ran them through LC-MS-MS.
Liquid chromatography tandem mass spectrometry.
Right. This technology separates and identifies molecules based on their mass and charge, which allowed the researchers to track 3,352 different microscopic metabolites.
We're talking about the vast sea of sugars, fats, amino acids, and cellular exhaust that basically dictates human life.
Yeah, so they mapped the comprehensive metabolic fingerprint of the donor pig before the surgery. Then they mapped the baseline metabolic fingerprint of the human recipient before the transplant.
And the crucial question was, after 106 hours of a pig liver running the human body, whose metabolic fingerprint would dominate?
You would intuitively think that because the liver is the primary metabolic engine of a mammal, the human blood chemistry would shift heavily toward the pig's baseline.
Right, that's what we expected.
But that isn't what the mass spectrometry showed. The post-transplant metabolic patterns strongly correlated with the human recipient's pre-transplant baseline.
It was undeniable evidence of cross-species metabolic reprogramming.
Wow.
The pig organs were pulling human substrates out of the bloodstream, running them through their porcine cellular machinery, and producing metabolic outputs that closely aligned with the human metabolic set points.
The organs were chemically conforming to their new environment.
Exactly. And they tracked specific sentinel molecules to prove this continuous adaptation. For instance, they monitored taurocolic acid in the bile.
Okay, and what happened there?
Over the 106 hours, the concentration of taurocolic acid progressively increased, carving out a trajectory that perfectly mirrored the metabolic recovery pattern of a stable human-to-human liver transplant.
Now, looking at this data, we do have to acknowledge the absolute boundary of this study, which concluded at 106 hours, just under 5 days.
Right.
Which raises the obvious question. If the organs were functioning this well, clearing creatinine, producing bile, metabolically adapting, why stop at day 5? Was there a sudden physiological crash?
No, actually, there was no biological failure. Terminating the observation at 106 hours was a strictly planned ethical boundary.
Right.
The medical team was operating with profound respect for the traditional views and customs of the decedent's family. The family generously consented to the study to advance human knowledge, but they requested a maximum observation period of 1 week.
And the team honored that commitment.
Completely.
That ethical clarity is vital. I mean, we owe this entire leap in medical science to that family's generosity. Are there other limitations in the data we need to consider?
Well, the primary scientific limitation is the lack of a direct control group. This exact procedure, orthotopic multi-organ xenotransplantation in a human, had never been done before.
Right, it's a first.
So when we see the metabolome shifting toward the human baseline, we have to recognize that some of those massive biochemical shifts could simply be the body's natural response to the extreme trauma of a multi-organ surgery.
Rather than like xenotransplantation-specific magic. Okay, final recap for the listeners. Bullet 1. Untargeted metabolomics. Tracked over 3,000 molecules.
Bullet 2. The pig organs adapted to the human body, shifting toward a human metabolic profile.
And bullet 3. The study safely concluded at 106 hours, proving technical feasibility.
Which leads to our final question for the listener. How close are we to seeing this in living patients?
Because even with those caveats, what this team proved is staggering. They didn't just show that you can physically connect the plumbing of a 6-gene edited pig liver and kidneys to a human.
Right.
They proved that those organs can survive human blood flow, evade the immediate destruction of hyperacute rejection, and actively regenerate tissue during an innate immune invasion.
Furthermore, they laid down a multi-omics roadmap. By using single-cell RNA sequencing and untargeted metabolomics, they've shown future researchers exactly which immune pathways like ADGRE need to be modulated.
Yeah.
And exactly how cross-species organs chemically adapt to human biology.
It fundamentally shifts the paradigm of transplant medicine. Right now, the entire system relies on human tragedy. We wait for someone to lose their life so their organs can save someone else.
It's a difficult reality.
But if this technology progresses, if we can manage that immune response past the 106-hour mark, organ transplantation becomes a scheduled, manageable medical procedure.
If your kidneys fail, the solution is just waiting for you in a specialized, gene-edited facility.
It moves xenotransplantation out of the realm of science fiction and places it firmly on the horizon of clinical reality.
It really does.
Which leaves us with a deeply philosophical thought based on everything we've explored today. I want you, the listener, to think about the ship of Theseus.
Oh, the ancient thought experiment.
Yeah, if you gradually replace every single wooden plank on a ship over time, is it still the same ship? Think about this multi-organ transplant at the microscopic level. If a liver from another species is placed inside your body and it begins to metabolize your unique human substrates, and it actively replaces its own circulating porcine albumin with your human protein isoform.
The assimilation we talked about.
Exactly. At what microscopic point does that organ stop being a pig organ and truly become yours?